Good morning, everybody. Today we will talk about gene expression in eukaryotes. Now, what is gene expression? Gene expression is the process by which information from a gene is used to synthesize functional gene products. And by gene products, I mean the proteins. Now, the ultimate effect of gene expression is that it will affect the phenotype of that particular individual. Now, irrespective of the complexity of the organism, whether the organism is unicellular or multicellular, every organism decides when and how a gene is supposed to be expressed. One example is your specialized cells. Now, your specialized cells, as in the cells in your heart and the cells of your liver, have different functions. Though the genetic makeup is the same, still they have different functions. This is because there are certain genes which is expressed in your liver cells which are not expressed in your heart cells, and it works vice versa also. There are even instances where in a particular cell a set of genes is functional only during a particular period of their lifetime. After that they're always turned off. Now all of this is because the gene expression is regulated. Now, like I told you, gene expression involves conversion of your DNA to your functional protein. So, gene expression occurs in basically four different steps. One is transcription, then you have transcriptional modification, then you have translation, then you have post-translational modification. So, the regulation of your gene can occur in either of these four steps, and the regulation, as in which step it occurs, depends on the genes. There are certain genes which is regulated at more than one level. So, let's look at each level in detail. Now coming to transcription. What is transcription? Transcription is the process of formation of all forms of RNA, be it mRNA, tRNA or rRNA, using DNA as a template. Now your transcription occurs in the nucleus. In case of eukaryotes, it is to be noted that the location of where the process occurs is important because in eukaryotes you have different cell organelles and you have a membrane-bound nucleus. So it is a little more complicated when you look at gene expression in eukaryotes as compared to prokaryotes, because in prokaryotes there's no cell organelles. So and in prokaryotes you have the operon system which is used to express gene expression. Okay, so coming back to transcription, like I told you, the gene can be expressed in four different levels, but mostly the genes is usually expressed at a transcriptional level. Now, transcription level, when you talk about, it is regulated because of the binding of your transcription factors. So, your transcription factors are nothing but proteins which can affect the transcription process. That is, it can either activate a transcription process of a particular gene or it can inhibit the transcription of a particular gene. And usually in case of eukaryotes, these transcriptional factors do not work alone. They work in combinatorial manner. That is, they work along with certain other protein which helps in the regulation of your transcriptions. So next step was RNA processing. Now the mRNA, that is the end of, when you look up, if you're looking in case of mRNA, the end of transcription is the formation of an mRNA. So like I was telling, so the mRNA what is formed after transcription is called as your pre-mRNA. Now there are reasons why this occurs. It is because usually the mRNA what is found in the nucleus, but then for translation your mRNA will have to move from the nucleus into the cytoplasm. So in cytoplasm there are certain enzymes which can degrade your mRNA, so your mRNA has to undergo processing. So there are three major steps which are involved in RNA processing, that is capping, tailing and splicing. So these steps involved conversion of your pre-mRNA to your mature mRNA. Now your gene expression can be regulated at either of these three steps. That is, it can be regulated during the capping process, during the tailing process or during the splicing process. Now splicing in general is removal of the introns and joining of the exons to give you a functional mRNA. But then some genes can be regulated by mechanism of alternative splicing. Alternative splicing is when the gene does not undergo the regular splicing. Instead, in some cases, instead of the introns being removed, you can have the exons being removed, or in some cases certain introns will be left in the gene. So the gene can be regulated at either tailing, capping or splicing, or it can be regulated by the mechanism of alternative splicing. The next the next step in gene expression is translation. Now, translation is the end, that is, it is the formation of a functional protein using mRNA as a transcript. Now, like I told you, your mRNA is already undergone processing because your translation occurs in the cytoplasm. Translation also, like transcription, had transcription factors binding which can affect, that is, which can activate or inhibit transcription process. Translation also has certain regulatory proteins which can bind to your mRNA, which can either activate or inhibit translation. The other mechanisms involved, the first one involves post-translation modification. The post-translational modification involves the post-translation modification is when so your protein which is formed at the end of translation need not always be a functional protein, because there are certain instances where these proteins undergo modifications like cutting and stuff so that they become functional. So depending on a protein, there are different steps involved in post-translational modification. So your gene can be expressed even at that post-translational modifications. The other two things on which gene expression depends is on two things. One is chromatin accessibility and one is mRNA stability. Like you know, chromatin accessibilities, your genes are not are in the form of a chromatin, which is a distinct feature when you compare to prokaryotes. That is, it can either be euchromatin or heterochromatin. So if it is euchromatin, it is transcriptionally active because the genes are placed a little far away from each other, whereas on heterochromatin, they're transcriptionally inactive because they're closely placed. So these also play a role in gene expression. The other one is mRNA stability. The half-life of the mRNA formed of a particular gene also plays a role in how this gene is being expressed. So ideally, your gene has to be gene expression has to be regulated for the normal functioning of a human being. Even if there's a small error in gene expression, then you can see it as a defect in the individuals. Thank you, and these are my references.